May 15th. So every other Wednesday at the same time, 3 p.m. Central. And today we're going to listen to Gina Guedira and Jared Smith lead a KASP workshop. So part of what they're going to cover is how to use KASP markers for genotyping, including some basic information about equipment, setting down and reading plates, and some discussion for strategies in designing KASP assays for SNPs and indels in wheat and barley. So Gina is the PI of the Eastern Regional Small Grains Genotyping Lab and has been in charge of that since 2004. Prior to that, she worked at the ARS in Manhattan, Kansas on germplasm enhancement in wheat. She got her PhD in genetics from Kansas State University, working with wheat, and prior to that got her master's degree at Davis working on barley. We'll also be hearing from Jared Smith, and he's a technician at the genotyping lab and has been there since 2005. His master's degree came from the University of Cincinnati, and prior to moving to Raleigh, he worked at the US EPA in Cincinnati, Ohio, genotyping not wheat or barley like the rest of us, but fish as an indicator species. So fun little tidbits. During this presentation, Gina and Jared won't be able to see the chat box since they're presenting using their full screens. But if you type in questions, I will read those out loud to them. Some questions you can personally email Gina at either of those email addresses. Her NCSU email address, Gina and _ is supposed to be included in that.
Thank you, Sarah. And you know, I was thinking, you may need to mute your microphone, Jared. You can do it. Okay. So I was thinking when I was getting the bio information ready for Sarah, that when I worked on my master's in barley, I used isozymes because that was the state of the art. And then when I was working on my PhD at K-State in wheat, you know, RFLPs were still the rage. And then when I started my position there, we were just starting with SSRs. So it's been a lot of progress and hopefully movement forward, obviously movement forward for both wheat and barley. And so, you know, Jared and I set this webinar up to sort of walk you through the process, assuming you're going to start using KASP markers. And so it's really oriented towards people who are in the lab using the markers in very specific things, you know, about plates and plate seals and reading plates and stuff. So I'll talk at first, and then Jared will be presenting some information, particularly on about how to do the plate reads and using the softwares. So I wanted to start out with emphasizing the fact that KASP assays are singleplex markers. That means you're running one marker at a time on a sample. And so with all the other technologies that we have, you know, why would we be interested in new types of singleplex markers? And, you know, we have a lot of potential and current usage of Illumina genotyping and also genotyping by sequencing, but in your own laboratories, generally people are not doing that themselves. And so you need a technology that can be used to do marker-assisted selection, but also training in a lot of cases for graduate students. And so the KASP is, I think, one potential technology that you could be looking at. We currently are using SSR and STS markers a lot for marker-assisted selection. And it's a little cumbersome, you know. SSR markers can be done on an automated capillary electrophoresis system, but it's going in and using the software to read this sort of data. It takes fairly skilled person oftentimes, and the workflow is kind of cumbersome. It's a lot of sample handling. And so we wanted to look for some kind of technology where we could be doing homogeneous assays. And a homogeneous assay just means that once you put the reaction in a well on a plate, you don't do any additional liquid handling. So it's a simplified workflow. In addition, oftentimes we want to be able to assay SNPs, but we have a number of cases where we need to be able to assay indels. So we want a technology that can be used for doing either one. In the genotyping labs, we need things to be suited to automation, but also to what we call boutique projects, where people send us, 16 samples and they want every marker you have for any trait done on those samples. And that may be the kind of thing that people are looking at doing in their own labs. And even some of the markers that we have, at least in wheat, for example, the normalization genes, currently they're done on gels. And we still wind up doing more gels than we would like to be doing. So these are examples of the different kinds of technology that are being used in each of the regional genotyping labs. The Fargo lab is set up to do Illumina SNP genotyping, and if on this graph, if you have numbers of samples on the y-axis and number of SNP on the x-axis, that technology is really suited for doing large numbers of markers per sample. And the same for genotyping by sequencing. The labs in Kansas and in Pullman, Washington, are both using the Sequenome system. And Sequenome is a technology that has some flexibility to it, but in terms of doing a single marker at a time, it's not probably the best choice. It's probably best suited for when you're needing to do a dozen or so markers on a sample. But there are two different types of homogeneous assays that we've looked at for using on a single marker at a time, or very few markers. And that is TACMAN, which is a very good technology, very reliable, and then also KASP. And so one of the things that we like about KASP is when we're looking at this graph, anywhere in this area, our cost per sample is the same. So the cost per data point really depends on the volume of the reaction you're doing. It's not depending on you're doing at least 1,500 samples, therefore something is cheaper. So we like that because there's a lot of flexibility there, but the bottom line cost is the same. So KASP is a chemistry that was developed by a company called K Biosciences, and it stands for Competitive Allele-Specific PCR. And so the K is there because K Biosciences. And they have been bought by LGC Genomics, and so they're now a part of that company. I'll probably use those two names interchangeably. But so if you see websites with, or if you see something with a link to a K Biosciences website or something, generally you won't be able to go there anymore. You're going to be redirected to an LGC Genomics link. And so the KASP chemistry is a homogeneous assay, as I mentioned, that's based on fluorescence, so there's no separation of your amplicons. It's scalable in that you can use the same protocols whether you're doing 96, 384, or 1536 plates. I don't, well, in the big labs where they are doing genotyping services using KASP, they use 1536 plates. We use either 96 or most of the time 384. Their literature says that you can use very small amounts of DNA, and we found that to be the case. We don't normally, I think, go down to 1 nanogram, particularly since wheat and barley have such large genomes. But we do things with as little as 5 nanograms of DNA. And they use a structurally modified polymerase, which helps in the allele-specific part of the PCR. And the big cost saving is this, that you don't have fluorescent labeled primers for each locus. So the fluorescent labeling is in the master mix, and so this has a big cost advantage. You can try an assay, and you didn't spend a bunch of money doing the fluorescent labeling on the primer. If it didn't work, you can move on to the next one. So you can get to resources about KASP on the LGC Genomics website, which I'm actually going to go ahead and open and just, I don't find it to be the easiest website in the world to navigate around. So I just wanted to kind of show it to you. So I'm at the link, actually, okay, that is how KASP works.
Sound?
And I wanted to point out that you can download the KASP manual here. And so the protocols for doing stuff are available in that manual. And a lot of the information that I'm going to go through today is available in the manual. And then also I was going to go ahead and show this animation, which is pretty good on how the assays actually work. So I'll just let us listen to that. Oh. Can you hear it? You can't hear it? There's no sound. So Sarah, are you getting sound? I'm going to pause for a second. So Sarah, are you getting sound? Okay, so it looks like you guys aren't getting sound. Okay, that's okay. I can go through a, let's look at this real quick. Okay, so you all are seeing this. Can make it full screen. Okay. That's okay. I'll just do it. So I will just go through this, talk you through it. So you can watch that animation sometime when you have time. And so their chemistry consists of this primer mix, the master mix, and obviously your template DNA when you're setting up reactions. And the master mix contains fret cassettes. And so these are oligos that have either a FAM, indicated here with the F, and for some reason they made this orange, but technically FAM fluoresces in the blue range, and hex, which is in the green range. And so these oligos are quenched in the master mix because there's a complementary quenching sequence. And then when you get your primers, each allele-specific primer will be, it'll have a sequence that you determine that leads up to the SNP. And so the SNP sits on the 3' end. So in this case, it's to interrogate a GT SNP, so the primers have the complementary C and A. And then on those primers, there's also a sequence that's complementary to, or that this fret cassette, it's actually not complementary to, it's the same sequence as this fret cassette in the master mix. So in the first round of PCR, your allele-specific primer will bind. If you have a match on the SNP, it will elongate. So what you've done then is made a strand of DNA that includes the tail sequence and the locus that you are interrogating. And then in the next round of PCR, the reverse primer will elongate, and then you make a complement of the tail. And then in subsequent rounds of PCR, what you're doing is you are increasing the amount of fluorescence because the FAM oligo on the quencher is no longer quenched. It's bind to your complementary tail sequence. And then in this case, hex, you're not getting fluorescence because that fluorochrome is quenched. And then you do reading of these on a plate reader or a real-time PCR instrument that's able to detect the fluorescence. So I think that's what I wanted to show you on their website, was to do the animation that you weren't able to hear, and then also show you where that manual is. Okay, so this is also in the manual. So this is a slide from LGC Genomics. Basically, obviously, when you're doing your setup, you're going to have your DNA, the master mix. So the master mix has everything you need. So it has these fret cassettes in it. It has the polymerases in there, the DNTPs, magnesium chloride, everything, as well as this ROX passive reference dye, which we'll talk a little more about when we talk about plate reading. And then your primer mix with your target-specific primers. So you go through regular PCR, and the thing that's different, of course, is the fluorescent reading and the SNP calling, which we'll talk a little bit actually about the process of doing the PCR, because there are just some things you need to be aware of that are probably different from the way samples are currently handled in your lab. At least it was for us. Okay, so first thing is to talk about equipment. So you can do the PCR on your regular thermal cyclers, which is, for me, one reason that the system is attractive. We have a whole lot of thermal cyclers in my lab, and so we were able to use those. And we more recently bought a hydrocycler, which is a hot water bath PCR. And that has the advantage, it's not constricted by plate format. So we can do 96, 384, or if we wanted 1536 using the same instrument. Or you can do the PCR on a real-time PCR instrument. I would suspect that most labs will do their reading using a real-time PCR instrument. Or we recently purchased a fret-capable plate reader. And the real-time PCR instruments have that advantage that they're readily accessible. Often if there's not one in a particular breeder's lab, there might be one down the hall. A lot of times, diagnostic labs use them, plant pathology labs. And And they're also good instruments for when you're doing assay evaluation and optimization because you can add PCR cycles as needed um, very easily. So examples, there we have a Roche LC480. ABI also makes a couple of different real-time instruments, and Biorad. Um, uh, Most of the things we're going to talk about today have to do with the Roche instrument because that's the one that we have in our lab and are familiar with. One drawback to the real-time instruments is that unless you have changeable blocks, you may be limited to either a 96 or 384 well format with a real-time instrument. So the plate reader has the advantage of not being constricted by plate format. But often it may be an additional cost, and you need a pretty high-end plate reader to use this technology. Ours was in the 48,000 range. I think there may be some that are less. We have the BMG Ferrostar, which is what was recommended by K Biosciences for doing KASP. One advantage of the plate reader is that they can use this ROX passive for reading for passive dye subtraction and normalizing samples, provided you have a software that will allow you to do that. And we use the cluster color software, and it allows us to do that. The other thing is the choice of the PCR plates becomes more important. And so actually, this is one thing that's a little different from how people are probably handling their samples, and that you just have to make sure that you use white colored plates that can be used for reading fluorescence. And if you're using a thermal cycler and a plate reader, then the plates need to be compatible. And you have to worry with the plate readers about things like warping of your PCR plates because that can make your readings be off. And the other thing is the seals for the plate need to be optically clear and not have any adhesive on them. And so, they're just little things that are different from the way people are probably handling samples now. The template DNA is also important. It doesn't have to be very concentrated. We use things generally in the range of 5 to 20 nanograms per microliter, but we often are not checking DNA concentrations. We use both DNA coming from kit extractions and SDS extractions. But we do find that the source of DNA in an experiment should probably be consistent, so we don't like to mix some samples that were isolated with SDS and are dirtier with things that have come from a kit extraction. It seems to mess up the clustering. And one thing to be careful about is the EDTA concentration in your samples. And so this is a concern if you're using elution buffers and you're doing a kit isolation where you don't really have any control over what the EDTA concentration is in that buffer. We use kits for extraction and use that DNA. We normally just dilute them 1-to-1 with water, and that seems to help with the amount of EDTA that's in those buffers. When we resuspend our DNA, we actually have it's a low concentration TE buffer that we use. KBio actually recommends that you use a tris buffer or you just use water. The master mix, I get this question a lot, and the master mix has to be purchased from LGC Genomics. It comes as a 2X reaction mix, and so it contains everything that you need, as we mentioned before. And we buy it in large quantities and aliquot it and freeze it. So it's sort of hard for me to give you an idea of what your cost for master mix is going to be because we do buy in large quantities. but we figure it cost us about 5 cents a microliter. And so generally if we're doing a 2 microliter reaction, then that means the master mix in that well cost 5 cents because it's a 2x master mix. So the assay mix that they refer to, they call this a 72x assay mix, and that's the primers. Typically, there are 3 primers, as you saw in the illustration of how KASP works, but you can design assays that are dominant and they only have 2 primers. So you're only amplifying one allele, and we have had to do that for a few things. Occasionally, you just don't have sequence from the other allele because it's deleted. Or you can develop assays that have 4 primers. So instead of having a common reverse, you can have a locus-specific, 2 locus-specific reverse primers. The primers can be purchased from IDT using basic purity. You'll have to mix them together in the appropriate ratios if you do it that way. You can also purchase from LGC Genomics. And the primers, in that case, come pre-mixed, and the assay is in a single tube, which we find to be an advantage for storage, and they also have a 2D barcode on them. When you order from LGC, you can provide them with a sequence, and they'll design a primer for you in silica and synthesize the primers. So that's the context sequence around the SNP, or you can provide them with primer sequences. They also do a thing, and that's called, when they do the in silica design, it's KASP by design. They also do a thing called KASP on demand. In that case, you provide the sequences and template DNA, and they'll actually validate the assay, and they're guaranteed to work. So if you have an assay for a gene that you really want to get it working and you don't have the expertise or the time to deal with validation, that might be an option. This is I'm kind of going through the sample flow in our lab. We always use 384 well plates, unless we're just validating a marker. But once we really start using it, we do it in 384 well plates, and that's because we can do small volume reactions. So a 384 well plate, we do a 2 microliter reaction, whereas in a 96 well plate, we do a 4 microliter reaction. So it costs twice as much to do the reaction in a 96 well plate. We have our master DNA plates. They're 96 well plate typically. Just things to consider about that is you're going to need a blank well when you run the samples. So you need a no template control. It's best also to have controls for your 2 different alleles. And so we tend to call things allele X, allele Y, and allele X. That's because you're reading a graph with a Y and an X axis. So it's just a function of the first software we started using. But having the control samples there is good for establishing your clusters and making sure things look okay. And then we dispense our DNA into the daughter plates. And KBio's protocol recommends that you dry your DNA down, and that's what we tend to do. And so you can dispense variable volumes to get a concentration that you might want in the reaction, because you're going to dry it down. So the amount you dispense doesn't really matter as long as the concentration is okay, or the final amount of dry DNA is okay. And once you dry the plates down, they can be stored at room temperature, and just not somewhere you're going to spill something into them. Or one other thing about that, it's one reason that we like
Can
to
you hear me,
do the
Sarah?
protocol where you dry the DNA
I
down is
should
it's
be
easier to
unmuted now.
pipette 2 microliters
Okay, can you
than
hear me?
1. So if we don't, if we
Okay,
use
so
1 microliter of DNA and 1
the
microliter of
the
master
last
mix,
step in this protocol, just picking up
it's a little
for
harder to
where
do the
from where
pipetting.
Gina left off, is actually doing
So
the
obviously the configuration
plate reads
depends
after
on
the
the
initial
project.
touchdown
We
PCR
find 24
program.
samples is really a
And what
minimum
that
for
involves
establishing
is taking what's
clusters.
called
So
an
you're not going to be able to
endpoint
do this just on
fluorescent
2
read
parents of a mapping
for
population,
at
as
least two
the only
appropriate
samples or
channels,
2
one
parents from
being
across.
the FAM channel,
And really having
the other
96
being the
samples is
hex
good
channel, which are the
for
two
your initial
fluorochromes
evaluation.
present in the KASP master mix.
Okay.
And there's also a
So
third
obviously adding
fluorochrome present in
you
there,
add
which
the
is the
reaction mix at a
passive
1x.
ROX dye, which, depending on whatever
It should
system
be
you're
1x
setting
in
up
the
in your
final
lab, you
reaction.
may or may not
They
be
say
able
your
to make
volumes can
use
go
of.
from 2 to 20. We've
I
never
just
done
wanted
anything bigger
to
than 8.
note that
Like I
these
said, we
given
do 4 microliters in
excitation
the
and
96 well plate and
emission
2
values
in
are,
a
they're kind of
384.
neighborhood values, and different
And then we have
instrumentation
always
may actually
initially,
go a
now
little
we
bit
use a
below
hydrocycler and we have to heat
485
seal our
and
plates.
a little bit below
But
520 or a little bit
for
above
the
520, and so
regular
on and so forth with the other
thermocyclers,
fluorochromes
we have a good
present
high
in
quality
the KASP
film that
master
we use
mix.
for sealing, or a good high quality seal, and it's
I
adhesive,
mentioned you
and
may or may
we
not be able to
continue
take
to
advantage
use
of the ROX
that and
passive dye. It
change
just
to an
depends on
optically
which type of
clear
software
seal before we
that you're
read,
using to do your
and that's
allelic
because we were worried about
discrimination.
evaporation
For example,
in
our
these small volume reactions.
RT-PCR instrument, the Roche
And so
480,
we felt like it was worthwhile to
does
um,
not allow us to
go ahead
do
and change the
passive
seals.
ROX subtraction automatically,
So
but the
for the
cluster caller
PCR, they recommend a
software
touchdown
that we
protocol,
obtained
read
from LGC
touchdown from
Genomics does allow us
67.
to use that
I might actually have that
passive
too
ROX
high.
subtraction feature.
65, I'm sorry.
Um,
Down to
so
57.
once
So
the
the
plate has
first
been
10
read
cycles are
the
dropping
first
down.
time, you're going to go through and
We
evaluate
tend
your
to go, they
clusters,
recommend 36 cycles. We tend to
perform
stop at
additional
33 and
PCR
start reading
cycles in
plates to avoid
3-cycle
overcycling.
increments.
And when
And
you do the reading,
so what I'm going to do at this
your
point is sort
it
of
should be done
orient
at
you
ambient
to
temperature, so the plate should not
what
be very hot or very cold.
what some ideal
And
output
like I
would
said,
look like,
we go
and
ahead and
just kind of
change the seals
recapitulate some
when
of
we're using
uh,
our Eppendorf
the features
machines for
of
doing
the
the
system that
PCR,
Gina has already touched on.
and you spin the
She
plates
had
prior to
mentioned that
reading.
data for
So I'm going to let Jared talk to
individual
you some
plates are
about
going
doing the
to be
reading
plotted
of
on
plates.
a X and Y axis, basically a
He
Cartesian coordinate system.
is the person who oversees this. One of the
Our
things that Sarah
Y-axis
didn't put in
is
his bio was
basically
our
the
KASP
axis which we're going to
guru.
see
Um,
hex intensity increasing as you
so
move
with
towards the
that, I'll
top
let him
of
show
the
you a few
graph
things.
here. And then on the x-axis, we have FAM intensity increasing out in this direction. Each one of these points of various colors on here represents an individual sample on a 384 well plate. And in effect, what we're doing in order to achieve allelic discrimination is just plotting those points based on using an ordered pair, if you will, of the respective hex and FAM intensities that were read by the plate reader or your RT-PCR machine. So if we just go through and look at the different clusters on here, there are 3 different clusters. What this represents is a codominant marker with that's segregating in a particular population. If we start down here at the XX cluster, these are going to be individuals which have a relatively high FAM intensity and a relatively low hex intensity. And then if we jump up here to the YY cluster, these are going to be individuals that have a relatively high hex intensity and a relatively low FAM intensity. And then the cluster here in the center is going to have an intermediate set of values for both hex and FAM intensities. So what these represent in this population are basically our two homozygous genotype clusters, being the YY homozygous for, say, an A, if it's an AC SNP, and then the XX cluster, which would be homozygous for the C, and then the XY cluster are going to be our heterozygotes, which would be AC. Another thing that I need to point out is the non-template controls, which ideally are going to show up down here at the origin of your Cartesian coordinate system, and you don't want to see your blanks. These will essentially be master mix with water added instead of DNA, or if you're using the dry down method, you would add the master mix and have to go back and add the water later to bring those particular wells up to a uniform reaction volume. Okay. I guess at this point I could point out the reason why we like this system so much is because this is a very intuitive way to go through and perform allele discrimination on a population. Gina had mentioned that, we sort of cut our teeth running SSR markers on capillary array sequencers in this lab. And, we did do some gel-based type work, and we make use of a lot of student labor in the lab. And I don't know how many of you out there who, have a lot of experience scoring SSR markers, marker data coming from a capillary array sequencer, but it can literally take years to get good at it. Doing allelic discrimination with your data in this format is very intuitive, and you can train student labor to be able to go through and do this discrimination extremely quickly. So this is one of the reasons why it's such an attractive system for us. Okay, so basically I'm just going to go through some of the different ways that you can actually perform your allelic discrimination depending on what sort of equipment you have in your lab. This first slide represents kind of the bare bones way of going through and doing it. Essentially what you have is some raw data output here from a plate reader. Going across from left to right, you have a position on the particular well that you're interrogating, your sample name, and then you have one column which represents data associated with the FAM fluorochrome, and the next column over represents data from the hex fluorochrome. Essentially, in a bare bones system, you could just plot these two data points for each sample into a basic scatter plot in Excel, and you should see some cluster formation occurring if you've got a good marker and you're doing everything correctly. This is the most rudimentary way that you could go through and do allelic discrimination, and it's not very efficient, and it doesn't offer the user a whole heck of a lot of control. Clicking on this point in a scatter plot really doesn't, tell you much information about what's going on over here, and subsequently you would have to go through and make a lot of manual calls, whether you're going to be YY or XX. If you had just a relatively low number of samples, you could potentially do that manually relatively quickly, but once you start getting into 96 and 384 samples, it's going to become way too cumbersome. So ideally, what you want to get your hands on is some dedicated analysis software for doing this type of allelic discrimination. I'm just showing a couple of examples here. This first one is a representation of the cluster caller data analysis software, which is developed and marketed by LGC Genomics, which is the company that has purchased K Bioscience. And then over here on the right, this is some instrument-specific software, which is associated with the Roche LC480 that we have in the lab. Most real-time PCR instruments nowadays, I think, will have some sort of endpoint fluorescence allelic discrimination software associated with them. The reason being, KASP has been around for a few years, but the TACMAN system has been around for even longer than that. And a lot of these companies, or a lot of people were using these companies' real-time PCR instruments to perform TACMAN assays. And in TACMAN, you're basically interrogating the same two fluorochromes. So, most good real-time PCR instrumentation is going to have this type of software for you. So at this point, I'm going to actually, oops, there we go. Thank you. So what I'm going to do is actually remote to a computer and show you just some of the features of these automated allele calling softwares. Let me get logged in here. Wow, right, first time. So I will just pull up some sort of pre-existing projects and show you what makes these programs so nice. Okay, so basically what this represents is some raw data taken on our light cycler 480. This is a pretty advanced cycle number. As you can see, there are already some pretty definitive clusters that exist. And once I go through this section of showing you, just some of the features of these different software types, I'll actually show you how we get to this point. But one nice feature of this software and of other softwares out there is it has an automated allelic discrimination feature where if I click calculate, it will automatically go through and assign individuals to either a hex or a FAM cluster. If you'll notice, we have the same setup with our Cartesian coordinate system here where we have increasing FAM intensity on this axis and increasing hex intensity on this axis. We've got some non-template controls down here. What's nice about this software is I can interact with these data points on this Cartesian coordinate system. For example, it looks like there are a couple of incorrect calls on my non-template controls down here near the origin. The software, wants to assign this to the hex cluster, but I know it's a blank. So what I can do is just quickly select those samples and then reassign them as blanks here. And so this is a lot more user-friendly than dealing with that cluster plot in Excel. So we'll minimize that. I'll bring up cluster caller, which is a standalone program, which means it's not really specific to a particular instrument. As I mentioned, this was developed by LGC Genomics specifically for people to do KASP allelic discrimination. They set this software up so that it would be compatible not only with a variety of plate readers, but also with a variety of RT-PCR output. So the way that I can go in here and show you some points of some allelic discrimination by importing a plate reader read. And again, this is an advanced cycle number. You can already see that the clusters are pretty well formed here, but there is a little bit of manual correction that I need to do here. So like with the Roche program where I could interact with these samples pretty quickly and reassign genotypes. These look like a couple of heterozygotes. I missed that one there. We missed this homozygous X. And then one of the neat features about this software is it will allow you to make use of the normalization using the passive ROX die. These clusters don't look so great right now. They're kind of strung out from the origin, which in my experience seems to be indicative of data generated by plate readers as opposed to real-time PCR instruments. But what I can do is go in here and normalize to the ROX passive die and tighten these clusters up. And I can actually go in here and normalize for crosstalk between the fluorophores and rotate clusters around their respective axes to give myself the tightest clusters possible. And then I can go back and unnormalize the data and make sure, it still makes sense. And allelic discrimination is performed that way. Again, these are going to be your YY homozygotes. These will be your XX homozygotes, and these are going to be the heterozygotes in this population. So moving on, I've put together a slide here which basically shows you the data collection workflow for for running the PCR and taking the endpoint reads, and then the decision tree that you're going to have to go through to determine, so is my data good enough, or do I need to keep cycling my plate in order to improve it? The first step is the initial touchdown PCR, and as Gina pointed out, these conditions can be found on the LGC Genomics website in their manual online there. They recommend a 36 cycle touchdown program. We have sort of adapted our protocol in our lab to 33 cycles, and I'll talk a little bit more about why we did that later. Once you've run your PCR, you're going to pull your plate off the thermocycler, stick it in the plate reader, and take your first fluorescent endpoint read. And then you're going to reach the first step in the decision tree. Do I have well-defined clusters? Are my controls that I put on my plate correct? If the answer is yes, then you can just export your data, perform your allelic discrimination and export your data, and you're done. If the answer is no, what happens is you will add an additional 3 cycles, take another endpoint read, and then ask the same questions of yourself. Do I have defined clusters? Are my controls in the correct clusters? Again, if you come to the conclusion that yes, I'm where I need to be, you do your discrimination and export. If you're still not where you need to be, you just repeat this step iteratively until you get to the point where you've got some good allelic discrimination and nice, well-defined clusters. So we can go through an example. This is just an assay for RHT B1, which is reduced height, assaying reduced height genes in the genome on the B genome. This is an example of 24 samples where I've taken my initial endpoint read. So I've done my 33 cycle PCR, taken that initial endpoint read. Again, we have the old familiar Cartesian coordinate system with FAM fluorescence increasing on the x-axis and hex increasing on the y-axis. At this point, there's really no discernible clusters here. You're just seeing a scatter of points. And you'll also notice the different colors of these points. These colors represent different genotypic calls in the allelic discrimination. It doesn't really make a whole lot of sense here. You know, you've got a couple greens here, a green here. You've got some pink, which represent unknown here and here, and then this red is a both alleles call here and here. This is basically random, and I'm going to need to recycle this plate. So these are the same data after adding 3 cycles and taking my second endpoint read. You can see at this point we do have some nascent clusters starting to form. You have this group of samples here, which is closely hugging the y-axis, and this group of samples here closely hugging the x-axis. We still have some imprecise calls in here. The sample here is, the software wants to call it an unknown, although, you know, it's pretty closely aligned with our YY genotypes over here. We've got another unknown down here, closely aligned with our XX genotypes. It's wanting to call this sample right here a heterozygote, which would be, you know, that's about the correct location for one, except for the fact that I know this happens to be in my non-template control, so I know this is not correct at this point. So what do? I add 3 more cycles and take yet another endpoint read. Now my clusters are pretty tight. Still got one unknown down here. My non-template control is now being called correctly as a blank. Most of these calls are correct. And if I add an additional 3 cycles, so at this point I'm 42 cycles total, I have pretty well defined clustering. My blank is where it's supposed to be. I would probably have, or I would definitely have a known positive and negative control, and I would expect to see them in their appropriate clusters here. So at this point, you know, I feel confident that I've made the correct allele calls, and then I would go ahead and export this data. So that's the basic workflow. You run the PCR, you take an initial endpoint read, you evaluate the clusters. If they aren't where they need to be, you add 3 cycles, you take an additional endpoint read, you evaluate the clusters, you ask yourself, am I where I need to be, so on and so forth, and you just repeat that process. Just a few things to keep in mind, and these are just, you know, nuggets of information we've picked up over the years running these assays. Sample prep does matter. You will notice that the presence of PCR inhibitors in your DNA extraction or your elution buffer can actually affect the total number of cycles you have to add to your samples to get good clustering. If you're using a buffer that contains 1x EDTA and you're running a 2 microliter reaction, you know, that's relatively a lot of EDTA compared to using a 1x TE solution where your DNA component is 2 microliters and the total volume is 10 microliters. If you're doing a 2 microliter reaction and your DNA component is 1 or 2 microliters dried down, then that's pretty high salt concentration. It's going to affect how many total cycles you're going to have to run to get good clustering. DNA concentration is another factor. The higher your concentration within reason, the faster you're going to get to your destination clusters. If you're running incredibly dilute DNA, it's going to have to, you'll probably have to add additional cycles than you would otherwise have to add. Auto scaling can play a big role in how good your data look. A lot of these programs are going to want to auto scale your data based on every data point on the plate that you're interrogating. If you have one weird well, for whatever reason, that has incredibly high background fluorescence, it could artificially crunch down the rest of your data points in what looks like a bunch of nulls, but if you actually have the capability to zoom into those data points, you might see that you have some really good clustering, and you just need to eliminate your outliers. Non-standard wells, again, if you're interrogating wells that have master mix and DNA in them, and you forgot to remove any blanks from your endpoint reads, they're going to artificially make your clusters look strange. And as a last resort, you can always go back in and evaluate your raw data and ask yourself, do they make sense? Allelic discrimination. The software is really nice that we use to perform allelic discrimination, but it's rarely 100% correct in making the proper allele calls. You're almost always going to have to go in and manually do some correction, as we saw in the example I gave earlier. Always be asking yourself, do the data make sense? You're much more intelligent than the allelic discrimination software, so if it's giving you really weird results and you're saying to yourself, well, this just doesn't look right, go back and evaluate the raw data and make your decision based on that, not necessarily on the auto calls that the software is making for you. And one point that I want to talk about in particular, sort of we undershoot our optimal cycle number, and we add cycles 3 at a time to get to our sort of optimum clustering. The reason why we do that is because there is a danger of overcycling these assays. That can result in the evolution of false positives. It can cause samples to shift from one cluster to the other. And it seems to be more problematic with dominant assays. And I can just quickly run through this example. This is we're interrogating for the TSN1 gene. It's a dominant assay, meaning we're going to have a null cluster for individuals where the TSN1 gene is not present. And then where the gene is present, we're going to see those individuals clustered up here in the YY cluster. This is what our clustering looks like at 33 cycles. This is our initial initial endpoint read after our touchdown PCR program. We already have pretty distinct separation. There are 13 individuals with the gene and 82.
So if people need to get to a meeting, they'll be able, or meeting or class or anything, they'll be able to watch the rest of it later if they want. Yep.
correct?
Mm-hmm.
It showed that these actually are the correct calls.
Yeah, so with the TS1 gene, that's the tan spot toxin sensitivity gene in wheat, and um, it's just not present in lines that are resistant to the toxin. So it's a deletion, and so we don't have any choice but to use a dominant assay. So it's not that there's a deletion in the gene, it's the entire gene is not present. And there are a couple of assays like that. The grain protein gene, the GPC, grain protein content gene on chromosome 6b would fall into that same category. But we also have some markers where there are null alleles. So we have an X allele, we have a Y allele, but there's a null allele. And so you do have a danger of overcycling those null alleles as well. So as Jared said, that's why we're running lots of different markers. And so that's why we tend to do things in what may seem like a longer step to get to our final, endpoint. I wanted to show you all, we're right at an hour now, Sarah. I'm happy to keep going. I have maybe 15 minutes worth of stuff. And I was kind of thinking, since it's recorded, if people need to leave, they could come back and look at it. Is that? Yes. Is that? Oh, somebody's shouting. Okay. Yeah. So I wanted to show you where you could get some markers you want to start using. And for wheat, there is a nice KASP marker resource available. There's a project at the University of Bristol that is been working on SNP identification, and they've decided to use KASP as their way of interrogating SNPs. So they've developed a lot of KASP primers, and so you can go to the Serials DB database and get access to these primers. So there are about 5,300 SNP that are available on the database now. I believe there are more than that, actually, that have been developed. And I'm going to go ahead and open that database. Okay, it came up here. I just wanted to show you, if you go to where it says here, Casper SNP database, there's a tab that says download all data. And so this includes the primer sequences, haplotypes in a set of European as well as some Semitic germplasm, and the sequences for the primers, the context sequences, and their blast hits. And just one thing I wanted to show you, they have noted these markers as dominant, partially codominant, and codominant. And so this has to do with the polyploid nature of wheat and compression of clusters when you are actually amplifying for more than one genome. So I suspect that the codominant markers are ones that are really genome specific. So if you look across their data sets for this codominant marker, they're either called C homozygotes or T homozygotes, whereas one where they've called it dominant, you see and now they're using the nucleotide as opposed to the XY that we generally report things as. So you're seeing a GT call, so this would be a both allele call, as well as things that are Gs or are, actually I'm not sure if there are any Ts in this row. I think what they're getting is just GT and G. So it's probably a single locus, but it may be that you are not able to call heterozygotes. And then over at the end of this file, they have the map locations. So I think that's probably kind of small on people's screens. The other thing I just wanted to show you here is you can also use this tab that's select primers, which I find to be real useful. So let's say you're wanting some markers on chromosome 5A. A map of 5A will show up. You can, if you're interested in this particular SNP, you can type in the ID here, or you can just click and download all of the markers on 5A. And so it comes in a nice, oh, there you go, in a nice Excel file that again has this map. And there are some microsatellites on these maps, so you could use them to anchor. There's a few dart markers. And then again, you have the SNP name, map location, and primer sequences here. So that we've used some of these markers. We've gone in and had particular projects where we wanted to enrich a QTL region with markers, and so we obtained some of these markers, and so they, excuse me, okay, and so they worked quite fine. The problem is that you don't, generally for U.S. germplasm, we don't know if they're polymorphic. And so if you are going to be using, so often people already have data for the 9K SNP projects from Illumina in wheat or from the barley Illumina assays. So you could, so where would you get sequences that you want to, I guess, convert, for lack of a better word, to a KASP assay? And so obviously would be from mapping in biparental map populations if you have SNP flanking your QTL, or you look at a consensus map and you look for a SNP in the region of a linked SSR or DART marker, or results from the association mapping. And so we've already done some work on things coming out of association mapping. I know particularly Mike Bumfries, the work in wheat on resistance to rust. They've worked on converting markers to KASP assays. The other one thing that approach that we used early on, because we didn't have map locations for the Illumina SNPs from the 9K, was we looked at LD for SSR or STS markers that were linked to the trait, or in some cases they're actually functional markers, so they're in the gene. And so the advantage there was no map position required technically for either marker. But we didn't have to, we just didn't have map positions initially. And there are some SNP on the wheat 9K that do not have map positions. So they were not polymorphic, or for whatever reason were not placed in the populations that were used for mapping. The other thing is you can use genotypes on lines that you've had done with the Illumina system, but you don't have phenotypic data on them. What you have is data on another marker that's being used to predict the phenotype. And so using that approach, we were able to get markers that we are currently using to detect the SR36 PM6 translocation on chromosome 2B. So we were able to identify SNPs that were specific to lines having SR36. The same for the 1RS, 1BL translocation, and the soil-borne mosaic virus resistance gene on the long arm of chromosome 5D. And in this case, is one of those cases where the SSR markers were not, or I'm sorry, where the SNP markers were not mapped, because the um, the density, marker density in the D genome is lower for the SNPs, and so they weren't able to put those markers on a linkage group. But by looking at linkage disequilibrium with the SSRs, we were able to identify SNPs that are actually diagnostic for the presence of the gene. One caveat to this that I would caution people about is that the LD is really specific to the germplasm that you assay. So you can have markers that are in good LD with a gene in your germplasm, but you can move into a different germplasm base and it'll be different. And that we found was the case for GLUD1 and the Amigo translocation, the 1RS, 1AL. When these markers work really well in our germplasm, but when we get out into more diverse germplasm, we see some discrepancies. And for the 1RS translocations, we were using the SCM9 marker, which is an SSR that's rye specific. And generally in this approach, we were taking things with the R-square really close to 1. But we have done some enrichment of a region where we took things at a lower R-square, and we were able to get markers that map in our general QTL region. Okay, so if you want, let's say you have these SNP from the Illumina Golden Gate or Infinium assay, you want to develop a SNP marker, the first thing you need is the sequence. Because generally the data that people have gotten is a locus by DNA table with AB calls. And so you can find the context sequence and the translocation at the T3 database. So I'll just show you how to get there. You can choose, you can go to the wheat or barley database. It works the same in both. There's a tab here that says download, and then there's something down here that says SNP alleles and sequences. And so these are the context sequences and the AB translocation to A, C, T, and G. And there we go. Okay, so the way this file is set up, it has the marker name. This is in barley, so it's the marker name on one of the barley OPA. There seem to be these synonyms for SNP names in barley. I'm not sure I'm real clear on all of them. This is if you got an A in your allele report, this tells you after the first comma what that A corresponds to in terms of nucleotide, and then the same for the B allele and the context sequence. In the barley file, you'll see the first of this data, the top of this file, the sequences are pretty long. And then you get into a section that's a little shorter, and I think the first SNPs in barley were from EST, so there's more sequence. And then you get into an area where these look to me like they were probably from 454 reads, or contigs that were built from shorter sequences. And then on down toward, if you go down further, you get into a section where the sequences are really short, and these are GBS tags. And so these are sequence tags that have been mapped in barley. This one is MB, which stands for the mapping population, and then has GBS. These are 64 base pairs of sequence. And the first few base pairs are complementary to the it's the PST1 cut site because you're always sequencing, from the same um, site for every sequence. And so KASP can actually be used to assay very small sequences, things as short as 50 base pair, because you don't need more than one nucleotide in between the forward and reverse primer. So it's possible that you could maybe use this information to develop a KASP assay if you identified something by GBS, but you really need the SNP to be right in the middle of the sequence. And this 64 base pair is just really related to the Tassel software. If you use a different pipeline, you can maintain longer sequences. But Tassel trims everything to 64 base pair because it's much faster in terms of computing because it uses less memory. So I think that's what I was going to show you in T3. Oh, there is a place in T3. Let's see if I can go back for just, well, there is a place there where you can blast a sequence against the marker database. And that way, if you had a sequence you were interested in, you could look and see if there's something already on the Illumina for it. Okay, so in Barley, we've done some transfer from the Illumina platform to KASP, and it was really straightforward, at least for the markers we've done so far. We had a recombinant inbred line mapping population from George Dukovsky, and we had screened it with the 384 Veracode OPA, and the OPA was not designed to be polymorphic in that population, so we only got probably less than 50% of the markers were polymorphic, and so we needed to fill in some gaps. And so George identified a polymorphic SNP. He sent us the sequences for 67. We just went in and designed primers for all of them, and 62 of them gave us usable data. So there was a 92% conversion rate. And of those, there were a couple that had compressed clusters, which would be the same kind of thing I was showing you in the database on the serials DB, where one of the homozygous clusters actually looks like a heterozygote. That makes it then difficult to score real heterozygotes. And this is a real population, and there weren't that many real heterozygotes in there. So it may be that this one works fine for scoring heterozygotes. We don't really know. And then 58 of them were nice codominant SNPs, and there were 2 that were actually polymorphic between the parents but didn't segregate in the population. So perhaps one of the parents or the actual parent used in making the cross was a heterozygote. This is just examples of the output with some of those markers. This is the output from Cluster Collar. This is a marker that worked very well. And so there are some heterozygotes in there, but we get very nice discrimination. And then 2 of them I recognized from the list as being genes that people might be interested in. One was the BURN3 gene. And so this one worked pretty well. I actually think that these maybe these 2 are a little bit questionable, whether they're really heterozygotes or they should be in the Y cluster. And then this is the CBF. So these are the genes that are involved as they're transcription factors for the core genes involved in cold tolerance. And you see these 4 clusters. And sometimes that happens. You see like multi-clusters, both with KASP and also with Illumina. And this is a known to be a multi-gene family. And so it may be that these clusters are related somehow to copy number, or to some residual SNP in a priming sequence in another copy. I'm not exactly sure. But we had flanking other markers in the region, and I could, once we looked at the allele report, we could tell that these should be considered Ys based on the flanking markers, and then these should be considered the other parent allele. There is also a project that I'm involved with on a collaborative development of markers, a marker-assisted toolbox of KASP assays for wheat. So these are markers that are very closely linked to, in most cases, diagnostic for the presence of a particular gene. And so this is an international collaboration involving Suzanne at the genotyping lab at CIMMIT in Mexico, Peter Sharp in Australia, a group of people in the UK, as well as Catherine Ravel at INRA in France. And so the idea is to have assays that can be publicly available. And we are willing to share these assays. Right now, they've not been published, and so you need to contact one of the collaborators to get the assays. One thing that I would ask for those people who are also developing assays that would have similar use would be to reciprocate in the sharing of those with the genotyping labs and with this group, so that they can be used as widely as possible in wheat breeding. And I'm not aware at this point of a similar effort to develop KASP markers for barley. I think it's a nice system that barley could really be looking at. So often we are just going into the literature and we're looking at published markers and we're assaying the same polymorphism. And that is much easier said than done in a lot of cases. A lot of times, for one thing, the polymorphisms aren't SNPs. So a lot of our functional polymorphism is based on indels, duplications, rearrangements, various things that are not just straight up SNPs. But the technology will allow us to assay both small indels and large ones. We haven't been, well, we have some assays that are based on duplication. Some of the duplications are really hard to deal with. A lot of times, so we're having to do some additional sequencing. So what you get is a publication, a development of an STS marker, which is great. It's a allele-specific marker based on a polymorphism in the priming sequence. But the fragments are a few hundred base pairs, and that's actually something that we haven't mentioned. Normally, we are targeting fragments less than 200 base pairs for amplification. And so when you're comparing a 600 base pair fragment with a 520 or a 450, those you can't just use the existing primers. So you have to go in and do some additional sequencing of the amplified fragments. In some cases, we need to do it from more diverse germplasm in order to have a really predictive polymorphism. Some STS markers are actually dominant. So you only get, even if you sequence the amplicon, you only get sequence from one allele. And so that has been a bit problematic as well. So you have to be kind of creative in figuring out ways to get sequence from the other allele. But currently, through this group, we've developed functional markers for a number of traits related to quality in wheat. The GLUD1 locus is the 2+12 and 5+10 alleles. The pin A and pin B associated with kernel hardness, green protein concentration. That was the gene um, cloned by George Zhukovsky. This is a deletion, or not a deletion, but it's not present in most hexaploid wheat. And then the red color genes on the A, B, and D genomes. LR34 is a cloned resistance gene, LR21 as well, and then the TS1 gene for sensitivity to tan spot tox A, the dwarfing genes, and then there are multiple alleles in each genome for the PPD1 and the VERN1. And we have assays for a number of alleles, but not all, it's not all encompassing just yet. And then we use linked markers that are quite predictive of a number of genes, including SR2, FHB1, and the soil-borne SBM1 gene. And then these others are all translocations. And so the link marker, for the most part, is diagnostic. So KAPS markers are a real good type of marker to go after for developing a KASP assay. And so this is just an example for the SR2 marker, CS SR2. The published protocol is amplification of a product that is then use a restriction enzyme, BSPH1. I don't know if you guys can, yeah, it looks like, because I can see Jared's screen back here, so I can look and see if it looks like things are showing up well. So we're assaying the same SNP. It's the SNP in the restriction site, so you either cut or don't. One thing, though, that we had to do was have the sequence of the entire fragment. So they were, this is a good example of where we've targeted a really small fragment, actually. I think this is only 61 base pairs. And then this is a case where we actually get a null allele. This is a marker-assisted selection project of my plants that are in the greenhouse, and they're BC1 plants, and this is a case where you do as I say, not as I do, because I don't have a Y control on here. And these individuals are all heterozygotes. This is a marker that we use a lot. But my recurrent parents were either allele X's or some of them actually were nulls. I wanted to give you an example of designing a KASP assay for a deletion, and this is a figure from David Laurie at the John Ennis Center, where they've done a lot of work on the photoperiod genes, and the very widely utilized photoperiod insensitive gene, which is the allele out of cyano 67. The insensitivity comes from a approximately 2 kb deletion in the promoter region. And so this is a really big deletion, but you're able to assay it using KASP by placing the primers one of the allele-specific primers here goes across the junction where the deletion is. And so in this string of sequence, the section in red is the deleted section. And they have placed their common primer is actually a forward primer, which is shown underlined over here. And then it's very close
Gina,
to where the deletion
really
starts. And for
informative and
assaying the allele in the lines that do not have the deletion, so the sensitive allele, they are using a primer that includes part of the region that's deleted in the insensitive individuals, plus one base pair upstream of where the deletion starts. And then the other primer is located further away, so this would pick up the insensitive allele. So all of this in red would be absent in those individuals. And this primer is also present in lines that are sensitive, but because it's more than 2 kb away, it doesn't amplify. So there are a number of different
Yeah, I bet you
strategies
might
that you
get
could
some
use to try
emails
to target these
later.
kinds of indels. And this shows you some amplification using that primer that marker actually works very well. Of course, in wheat, we have to think about genome specificity. So this is an example of where we're designing an assay typical to any other time you're trying to get a genome-specific assay in wheat. In this case, it's the Vern B1 locus, the winter version of Vern B1, where we have aligned